Okay, everybody, can you guys hear me? Okay. So, we finished lipogenesis, right, and triglyceride synthesis last uh, Tuesday. And today we started talking about the catabolic pathways, like lipid oxidation, right? Fatty acid oxidation. Well, actually, we should also include triglyceride mobilization, right? To break down triglyceride to glycerol and fatty acids and then the fatty acids can be oxidized in the different tissues, and also ketogenesis. Um, so actually the catabolic metabolism of lipids should include triglyceride breakdown, or we also call lipolysis pathway, right? Or triglyceride mobilization. And secondly, the fatty acids oxidation. And third, lastly, the ketogenesis, right? Three catabolic metabolism pathway in the lipid metabolism, right? So now let's talk about mobilization of triglyceride. And before we talk about mobilization, I just wanted to mention you again the storage of triglyceride. Remember, this is pathway that occur in the fat condition, right? We try to store excess energy in the form of triglyceride in the adipose tissue. So this just represent the fat cells, right? So fat cell is actually, if you look at under fat cell under the microscope and compare to other different type of cells, fat cell is very different and special. I mean, the fat cell size is much bigger and filled with lipids, right, with oil. So this type of cells store basically triglyceride. And this just represent the mitochondria in the fat cells. So in the fat condition, when we eat food and we absorb dietary fatty acids, right? Dietary fatty acids, and if you still remember, the fatty acid once it's enter the enterocytes and need to be re-acidified again, right, to the triglyceride. And the purpose of this is to help our body to move lipids or fatty acids from the intestine to the adipose tissue for storage. And again, I also mentioned that lipoprotein is very important that help transporting fatty acids, right, or triglyceride from one tissue to the other tissue. So the chylomicron is one of the lipoprotein that is made in the small intestine, actually packed the triglyceride and transported the triglyceride from intestine to the adipose tissue for storage. And before, again, triglyceride can be taken up by fat cells, triglyceride need to be broken down again, right, into fatty acids and glycerol. So fatty acids are the molecules that are able, that can be taken by fat cells. So once fatty acids enter the fat cells, and three molecules of fatty acids together with one molecule of glycerol 3-phosphate, right, for the synthesis of triglyceride that we talk about, right, last class. And And before fatty acids can be used for synthesis of triglyceride, fatty acids need to be activated, right? Remember? So activated to fatty acyl-CoA by an enzyme called a fatty acyl synthetase. So this is sort of, the storage of fatty for triglyceride that occur in the fat condition. Question? Yeah.
Repeat what the enzyme glycotrificase goes.
Sorry,
The last enzyme that you mentioned.
the enzyme to activate fatty acids, the fatty acyl synthetase, right?
Okay.
Synthetase, I'm sorry, synthetase. So, and in addition to fatty acids that initially from the diet, and liver can actually also synthesize fatty acids through the lipogenesis pathway. Remember, in the fat condition, right? We have extra energy, extra, glucose and protein that can be converted to the fatty acids. So lipogenesis also occur in the liver in the fat condition that can also make fatty acids, and this fatty acid can also be transported to the adipose tissue, right? Adipocyte. So again, similarly, triglyceride has to be packed into a different lipoprotein, right? So liver makes a different lipoprotein called VLDL, very low density lipoprotein, so that can pack triglyceride that synthesized in the liver and transport it, to the adipose tissue. So similar process, triglyceride either in VLDL or in the chylomicron eventually need to be break down to fatty acids, and that can be uptake by fat cells, right? So this sort of the whole process. And also we know that diet also contain carbohydrate, right? So we also take the glucose. So fat cells also able to take up glucose and metabolize glucose through the glycolysis pathway. And this uptake process is stimulated by insulin, right? If you still remember, we all talked about this before, right? So glucose, once it's enter the fat cells and go to the glycolytic pathway to make glycerol backbone, glycerol 3 phosphate, right? So this pathway is a major pathway for glycerol 3 phosphate synthesis in the fat cells. And so the other pathway, for example, glucose can of course be oxidized, right, in the mitochondria TC cycle, and the acetyl-CoA can also be used for lipogenesis in the adipose tissue in the fat condition. So and again, the liver does more lipogenesis, right, than the adipose tissue, but adipose tissue is also able to do the lipogenesis. So this sort of the lipogenic pathway, right? Fatty acids converted from glucose, or actually amino acids. So amino acid, I think I missed L. Oh, okay. So amino acids can also contribute to lipogenesis, right? And also pyruvate, if you still remember that when we talk about anaeroprosis or ketoprosis, right? The pyruvate can also be used to make glycerol phosphate. Any question about this whole process? I think it is pretty comprehensive, right? To try to connect all the, lipid and protein and carbohydrate metabolism together. So, And here I just want to point out that the enzyme that break down triglyceride to fatty acid glycerol is the lipoprotein lipase. We're going to talk about more about this enzyme. Just wanted to, lipoprotein lipase, LPL, right? Okay. All right, so now mobilization triglyceride, and this is basically the catabolic pathway that occur during the fasting condition, right? When we undergo the starvation and we don't have much glucose, right, obtained from the diet, so our body need to break down the stored energy and use stored energy. Of course, the breakdown glycogen in the liver is important pathway, right, to release glucose, to try to maintain glucose levels and also provide energy for other tissue use. And the other important alternative fuel our body uses during the fasting conditions is fatty acids, right? So fatty acids need to be released from fat tissue, which is the storage site, right, triglyceride. So and then provide energy source for the multiple tissues. So this just gives you kind of whole, the overall pictures. And in addition to fatty acids, the ketone bodies are also very important alternative energy fuels that our body uses during the fasting conditions. And the ketone body basically is made from fatty acid oxidation, right, and in the liver. So liver makes ketone bodies from acetyl-CoA, which primarily generated from fatty acid oxidation. We're going to talk about later, right? And so ketone bodies released from the liver into circulation and transported to the other tissue, particularly to the brain and muscle for use as energy, right? So again, during the fasting condition, in addition to break down the glycogen to release glucose, and liver can also make glucose, right, through gluconeogenesis. And in addition to the glucose, and our body also utilize fatty acids and ketone bodies, and of course amino acids, right, and as an alternative energy fuels. So this gives you kind of overall pictures, right, how our body, energy metabolized is regulated during the fasting condition. And then we can go to the details and see how triglyceride is breakdown and to release fatty acids. So this process is called a lipolysis, right, or mobilization of triglyceride. Any questions about this? Yes?
Where does the process start? Where does it all kind of start off?
Oh, how the body started this kind of pathway, you mean, right? Is that your question?
Yeah, so you're in the fasted state.
Right.
Which organs are going to respond first?
That's a good question. So I think the hormone levels change are the first, the drivers, like we talk about during the fat condition, right? You change the glucose level, so you change the hormone levels. And similarly, during the fasting, you also change the glucose level, right? You have a lower glucose level, and then you have a lower insulin secretion. So under the fasting conditions, you have a decreased insulin levels, and meantime you also stimulate the glucagon and epinephrine hormones release, right? So, and this hormonal levels change that are the most important regulators of all this metabolic pathway that we're going to talk about later, right? So, for example, the insulin basically suppresses lipolysis, so when we have a lower insulin level, then lipolysis suppression will be released, right? So you have a promoter. Oh, so sorry. Ah. I don't know. Oh, okay. Well, yeah, just did it. So, all right. What I talk about? Okay, so yeah, insulin suppression should be released during the fasting, right? And also we have increased the epinephrine level. So epinephrine basically stimulates lipolysis, stimulates triglyceride breakdown, right? And by stimulate the key enzyme that we're going to talk about later, right? For the triglyceride breakdown. That's a good question. Thank you. Okay, so mobilization in the fasted condition. This again is a fat cell, and we have a triglyceride, right? Store a lot of triglycerides stored in the fat cell, and during the fasting condition, the triglyceride will be breakdown, and then so the key enzyme for triglyceride breakdown is the HSL, hormone-sensitive lipase, right? Hormone-sensitive lipase. So this is the key enzyme that regulates triglyceride breakdown, right? Or regulates triglyceride mobilization, or we also call this process lipolysis, right? And as I just said that during the fasting condition, the epinephrine and glucagon levels are increased, right? Insulin levels are decreased. So epinephrine, glucagon basically are the catabolic hormones, right? So can stimulate cyclo AMP increase. Again, I just wanted to remind you that cyclo AMP basically is related to the catabolic metabolism, right? So epinephrine, glucagon stimulate increase cyclo AMP levels, which can causing the protein kinase A activation, and eventually can stimulate HSL phosphorylation and stimulate HSL activity. So HSL activity or hormone-sensitive lipase activities increased by epinephrine and glucagon, right, during the fasting. So then can promote triglyceride hydrolysis or triglyceride breakdown. And also because of a glucose levels decrease, right, during the fasting, so insulin goes low, and insulin basically suppresses HSL activity, right? So by dephosphorylated HSL. So if insulin levels low, that also makes HSL activities high, right? So this set of hormone, the hormone profile changes that promotes lipolysis, promotes triglyceride breakdown by stimulating HSL activity. Right? Does that make sense? Okay. Yeah. Question.
What did you say PK stands for?
Oh, protein kinase A is also protein that catalyzes phosphorylation of HSL. Yes.
So insulin, you said suppresses HSL?
Yeah, HSL activity is right.
But then,
Because you have a low insulin, so this suppression will be less, right?
oh, so it'll actually...
So actually, yeah, right. So this, just tell us how triglyceride is broke down into fatty acid glycerol during the fasting condition because of hormonal, level change, right? And during the fasting, this fatty acids are released from fat tissue are used by other tissues like muscle and other non-fat tissues, right, for energy. And also fatty acids goes to the liver. Again, I mentioned, right, for making ketone bodies. So liver is the primary site for ketogenesis, right? So ketone bodies, then once it's made in the liver and then released into the circulation and picked up by other tissues, primarily brain and muscle, and also used for energy, right? So during the fasting conditions, in terms of lipid metabolism, the triglyceride actually break down and contribute, to fatty acids and ketone bodies, like contribute, the alternative energy fuels. And glycerol, I actually forgot to put it. We should put an arrow here. So glycerol also being released, right, from the adipose tissue, and this glycerol usually is picked up by the liver for making glucose, right? That gluconeogenesis during fasting. So so just wanted to mention to you that although we're talking about lipid metabolism here, but you also wanted to connect, the glucose metabolism. That also happening as well, right? Yes.
So when you said that the HSL hormone-sensitive lipase
Right.
triglyceride mobilization.
Yeah.
So mobilization to where? To the adipose
Triglyceride mobilization. I mean, we moved from fat tissue, right, and release fatty acids, glycerol, and fatty acid glycerol picked up by other tissues and used for energy, right? Okay, so this whole process will also have a different terminology called lipolysis, right? So lipolysis is basically triglyceride breakdown, and also sometimes we call it mobilization of triglyceride, right? Any questions so far? Okay. Okay, now let's go to a little bit more detail about the hydrolysis or lipolysis process. Remember, triglyceride molecules, right, has a glycerol backbone and 3 fatty acids, right, attached to the backbone. So although I just mentioned that HSL is a key enzyme for lipolysis, but we also have other enzyme lipase that also involved in the lipolysis, like ATGL, adipose triglyceride lipase, right? So HSL is very old enzyme that was discovered a long time ago, right? And ATGL, adipose tissue triglyceride lipase, is discovered relatively recently, right? So we understand more, how the lipolysis is regulated. So basically, we have 3 enzyme: ATGL, adipose tissue triglyceride lipase, and hormone-sensitive lipase, and monoglycerol lipase, right? So 3 lipases that are involved in the lipolysis in the lipolysis process. Yes.
Is HSL sensitive to only insulin, or is it sensitive to glucagon?
Yeah, epinephrine glucagon is a major regulator of HSL, right? So triglyceride have 3 fatty acids, right, attached. So ATGL actually is the first lipase that catalyze the removal of the first fatty acids from the triglyceride. So, and generating diacylglyceride, right? You see, you still have 2 fatty acids attached to the glycerol backbone, and 1 fatty acid is being removed from the SN1 position. And the second step is that HSL. So HSL also catalyzes the remove of fatty acids from SN3 position, right? So generating the monoacylglycerol. And then lastly, the monoacylglycerol lipase that remove the last fatty acid from the SN2 position and release the glycerol backbone and the 1 free fatty acids, right? Yeah.
Is the HSL the most important? Like if we were going to describe the sequence of utilization, like if we go through the ATGL first, since it's the triglyceride,
Right.
or
Yeah.
the other diagram only mentions HSL?
Well, I mean, because HSL is more like regulated by the hormones, the more during the fasting condition. So this is, well, I mean, another explanation would be HSL may be more, understand by, the researchers.
So the other ones aren't hormonally regulated?
Well, ATGL recently people also found that can be regulated, during the fasting, but compared to HSL, it's less known, in terms of this enzyme. But I mean, for this class, we just, we just wanted to focusing on the HSL as a key enzyme. But I just want you also aware of, the other 2 enzymes is also involved in the lipolysis. Yes.
So if you don't have ATGL isn't working, HSL can still like cleave the SN1
Yeah.
and SN3, right? So
Right.
if you don't have ATGL, it's not a
Right.
problem.
Right. That's true. Yeah. That's good point. Yes. So you're still able to remove fatty acids, right? I mean, so make available fatty acids. So this all 3 enzymes work together, and to release fatty acids from glycerol backbone. So, and here we just talk about the specific each, the enzyme and how they do in the lipolysis. So, for example, ATGL primarily, the substrate for ATGL is triglyceride, right? So, remove the first fatty acids from SN1 or SN3 position, right? So, HSL is able to remove fatty acids from either SN1 or SN3 position, right? So, and producing the diacylglycerol and a 1 free fatty acid. And hormone-sensitive lipase, basically diacylglycerol as the substrate, and basically they remove, again, either SN1 or SN2, right? So generating monoacylglycerol with 1 fatty acid attached to the SN2 position in the middle. So again, both ATGL and HSL can cleave fatty acids from either SN1 or SN3 position, right? And the remaining fatty acids at SN2 position. So the last enzyme that cleaves the last fatty acids from the middle of the SN2 position is the monoacylglycerol. And there are actually also another enzyme called triglyceride hydrolase. It's also expressed in the fat tissue, but less effective compared to HSL, so that people usually don't pay much attention to this hydrolase in terms of its role in the triglyceride mobilization or fatty acid mobilization, right? So this is the last enzyme, right? Monoacylglycerol lipase, and basically remove the last fatty acids from SN2 position. So this, I just put all the three steps together, right? The HSL, ATGL work together to remove SN1, SN3 fatty acids, and then last monoacylglycerol lipase and remove the last fatty acids. So eventually, three enzymes work together and generating three fatty acids and one glycerol molecule. Okay, so transport. So once this fatty acid is released from triglyceride in the fat tissue, fat cells, and then released into the circulation, and in the circulation, fatty acids usually binds to albumin. As fatty acids usually cannot be just free molecules, in the circulation. It cannot be because it's lipids, right? It's the water, hydrophobic. So basically, need to be bound to the protein. So albumin is the most important protein that can bind fatty acids and help fatty acids traveling, right? So about 2 to 3 fatty acids, one albumin molecules can bind 2 to 3 fatty acids, right? And in terms of destination of the fatty acids, once it's released from fat tissue, 2/3 of fatty acids released from fat tissue are transported to muscle. So muscle is really the most important tissue that pick up fatty acids and utilize as energy, right? And the 1/3 of fatty acids that are released from fat tissue during the fasting condition goes to liver for fatty acid oxidation and ketogenesis, right, for making ketone bodies. And glycerol released from fat cell and also transported to the liver for gluconeogenesis during the fasting, right? Does that make sense? So they all, can connect together. We're going to do this later for informal writing, right? Now, any questions? Is that clear about triglyceride mobilization or lipolysis? Okay, so now let's talk about the fatty acid oxidation. So once fatty acids are released from fat tissue and picked up by other tissue, for example, muscle, right? Muscle takes up 2/3 of the fatty acids, right? And then utilize or oxidize fatty acids and for energy, for production. So now we talk about fatty acids oxidation. How fatty acids oxidize in the tissues and for making ATPs. Again, when fatty acids is oxidized, and fatty acids first need to be activated, like triglyceride synthesis, remember, right? So this is actually the same reaction for both triglyceride synthesis and fatty acid oxidation. So fatty acids need to be activated to fatty acyl-CoA, and by the same enzyme called fatty acyl-CoA synthetase, right? And this reaction utilize the ATP, right? From one ATP to AMP, that equivalent to like 2 ATPs, right? So basically, in this reaction, the 2 ATPs are used because the ATP is converted not ADP but AMP. That means that two, equivalent energy to the 2 ATPs are used in this process, right? And this reaction occurs in the cytosol, right? In the cytosol, remember. And I don't want to go to this. We already talked about before, right? The different numbering system. Now oxidation. So beta oxidation. And this process occurs in the mitochondria, right? So fatty acids activation to the fatty acyl-CoA occurs in the cytosol, right? And the fatty acids oxidation occurs in the mitochondria. So there are also some process, transport process needed to transport fatty acyl-CoA from cytosol into mitochondria for oxidation. So let's talk about oxidation first. So beta oxidation means that the fatty acids can be oxidized by multiple steps. That each step just go the similar reactions and cleave the fatty acids 2 carbon unit at a time, right? So this is called beta oxidation because each time when fatty acid being cleaved, almost basically at the beta carbon position. So this is called a beta oxidation. So and release the 2 carbon unit, which is acetyl-CoA, right? So and fatty acid oxidation also goes the cycles. So each cycle you release the 2 carbon unit acetyl-CoA, and then 1 fatty acyl-CoA with 2 carbon shorter, right? And the fatty acyl-CoA. So for example, palmitate, which is 16 carbon fatty acids. So when we go to the 1 cycles and we remove the 2 carbon unit acetyl-CoA from the palmitate and generating 14 carbon fatty acyl-CoA and a 2 carbon acetyl-CoA, right? So and we have palmitate has 16 carbons that eventually, when complete oxidation of 1 palmitate molecules, will generate how many acetyl-CoA? 8, right? So 8 acetyl-CoA will be generated, and how many cycles that we need to do to completely break down the fatty acids molecules, palmitate molecules? 7, right? 7. So 7 cycles, you need to do and to break down all the 16 carbon fatty acids to 8 molecules of acetyl-CoA, right? So again, this process occurs in the mitochondria. So I just mentioned that fatty acyl-CoA be synthesized from or being activated, right? Fatty acids being activated as fatty acyl-CoA in the cytosol and beta oxidation in the mitochondria. So fatty acyl-CoA needs to be transported into the mitochondria across the mitochondrial membrane. So there are actually 4 different enzymes that are localized in the mitochondrial membrane, inner or and out mitochondrial membrane. And all these 4 enzymes work together to transport fatty acyl-CoA from cytosol into the mitochondria. And here I would just want you guys to remember one key enzyme, right, which is highlighting red, the carnitine palmitoyl transferase 1, CPT1, all right? So CPT1 is the key enzyme that controls fatty acid oxidation, so basically control the transport of fatty acyl-CoA from cytosol into the mitochondria, right? So which makes sense, right? If you can control transport, then you can control the rate of oxidation. So the process would be so like in the outer mitochondrial membrane, there are two enzymes. One is acyl-CoA synthetase, and the CPT1 carnitine palmitoyl transferase. And these 2 enzymes are localized in the out mitochondrial membrane, right? And this inner mitochondrial membrane, this is the mitochondrial matrix, right, inside the mitochondria. So, and this space we just represent like cytosol, right? So once the fatty acids is activated to fatty acyl-CoA in the cytosol by the fatty acyl-CoA synthetase, and then acyl-CoA being entering the inner space, right, the mitochondrial inner space between outer and inner membrane, and the CPT1 then does the function to catalyze the formation of acyl-carnitine from acyl-CoA, fatty acyl-CoA and carnitine, right? So acyl-carnitine is the molecule that can be transported across the inner mitochondria into the mitochondrial matrix, right? So so one acyl-carnitine is in and one carnitine molecule is out. So this exchange, transport system catalyzed by a second enzyme called carnitine palmitoyl transferase 2, right, which localizes the inner mitochondrial membrane. And then, so eventually acyl-carnitine enter the inner mitochondrial membrane and can be converted back to acyl-CoA and release the carnitine. And acyl-CoA eventually go to the beta oxidation pathway, right? And carnitine can be recycled, to help the another acyl-carnitine molecules right, into the mitochondrial matrix. So just try to remember, there is a transporter system that help fatty acyl-CoA enter the mitochondria for beta oxidation, and there is a key enzyme in this system, the CPT1, right, that can control the rate of fatty acyl-CoA transport into the mitochondria. Any questions? That clear? Yes. Yeah. Say that again. Right. Yeah. No, no. So this acyl carnitine, right, converted back to uh, acyl-CoA by this enzyme, right? So acyl-CoA then enter the beta oxidation pathway and for finally be oxidized to acetyl-CoA, right? Anyway, acyl-CoA is a long chain fatty acids and acetyl-CoA is just a 2-carbon small molecule, right? Any other questions? Right? Okay, so let's take a break. Okay. All right, let's come back. So, okay, so I guess this diagram gives you a like whole picture starting from fatty acids to complete oxidation, right? A fatty acid into CO2 water, right? I know it's kind of small, but this process, right? The fatty acids, again, first of all, need to be activated to fatty acyl-CoA, and in the cytosol, right? And it's a mitochondrial membrane, inner, outer and inner mitochondrial membrane, and the enzymes are required for transport of acyl-CoA from cytosol into mitochondria. Again, we mentioned the CPT1, right? It's a key enzyme that controls the transport of acyl-CoA. And inside of mitochondria, beta oxidation occurs. So several reactions, right? 1, 2, 3, 4. 4 reactions. You can see the first is a dehydrogenation of acyl-CoA, right? So basically remove the 2 hydrogen from acyl-CoA molecules and forming delta trans eno-CoA, right? And also generating 1 FADH, reducing equilibrium. And then the second, you don't have to remember the enzyme, right? Catalyze this specific reaction, but catalyze this reaction is called acyl-CoA dehydrogenase. And then the second reaction is that additional water to saturate the double bond. So you can see here actually double bond is generated right after first reaction, the dehydrogenation. Then the next second reaction is adding the water to remove double bond or to saturate double bonds and forming hydroxyl acyl-CoA. And the next step is dehydrogenation again, and you're also adding remove the 2 hydrogen from the molecules and transfer the hydrogen to NAD, right, and generating NADH equivalent molecules. And the product of this reaction is the keto acyl-CoA. And the final reaction is the cleavage of the acyl-CoA molecules from the cleavage of acetyl-CoA molecules, right, from the acyl-CoA molecule. And the enzyme catalyzes reaction is a silase. And so you can see 1 cycles and you remove 2 carbon molecules, which is acetyl-CoA, right? And 2 carbon shorter fatty acyl-CoA with the 2 carbon shorter. And once and also goes this cycle for depend on the length of the fatty acids, right? I mean, if we're thinking about the palmitate 16 carbons, and this cycle will go 7 times, right? And to completely break down the fatty acyl-CoA molecules into the 8 molecules of acetyl-CoA, right? And eventually, as all the 8 acetyl-CoA molecules enter the TCA cycle for complete oxidation to the CO2 and water, and of course produce the ATP and again NADH, FADH, right? So just wanted to remind you here that in the 1 cycle of beta oxidation, we generated 2, actually 1 FADH and 1 NADH, right? This is kind of details. Again, this is the first activation of fatty acids. Any question? Right?
You had mentioned something about the enzymes, but I just didn't catch it.
Sorry?
You had mentioned something about the enzymes, but I didn't quite catch it. You said
Y-
that we needed to know all the enzymes or just the important points of generating NADH and
Oh,
ADP?
you mean the enzymes?
Yes.
Well, I mean, you probably don't need to remember all the enzymes.
That the
But-
what is generated and
Right,
what
right,
is placed.
right. Just to understand how the acetyl-CoA molecule is being removed from the acyl-CoA molecule. That goes several steps, right? I mean, dehydrogenation and addition water and dehydrogenation again. So, activation of fatty acid to fatty acyl-CoA and transport of fatty acyl-CoA into mitochondria. And this is the first reaction, right? You just mentioned, the dehydrogenation, right? You remove the two hydrogen from the acyl-CoA molecule and transfer two hydrogen to FAD and forming FADH, right? And it's just the rest of the reaction, right? Then adding water to satellite double bonds, and then the dehydrogenation again, remove two hydrogens and transfer two hydrogen to NAD and to generate NADH, right? Eventually you release the acetyl-CoA from acyl-CoA molecules, right? And it's just the overall reactions, the oxidation or the dehydrogenation reaction, right? This enzyme and this oxidation reaction basically you generating the reducing equivalents, right? And hydration, oxidation again, and cleavage. So basically 4 steps, right, in each beta oxidation cycle for making one acetyl-CoA molecule. So the question here, why more energies from fat than carbohydrate? We mentioned this before, right? And we make a comparison to how much energy that made from oxidation of 1 gram of fat versus 1 gram of carbohydrate. We are able to produce 9 kcal from 1 gram of fat versus only 4 kcal from 1 gram of carbohydrate. So by calculating the ATPs, that probably will help us to understand more, right? So in fat oxidation, I just talked about, right? The every loss of a 2 carbon or every removal of the 2 carbon as acetyl-CoA one cycle, right, and produces 1 FADH and 1 NADH, right? And each FADH we know can produce 2 ATPs, right? 1 NADH can produce 3 ATPs. Remember, this FAD and ADH are all generated in the mitochondria, right? So don't need to, go that different shuttle. So always, NADH always produce 3 ATPs in the mitochondria once it's made in the mitochondria. And we take the palmitate as the example. So palmitate basically is 16 carbons, right? So be able to produce 8 acetyl-CoA molecules, right? Once it's complete oxidized in the beta oxidation process. So and we know each cycles we mentioned earlier, right? This for complete oxidation of palmitate in the beta oxidation, we need 7 cycles, right? So each cycle produces 2 ATP from FADH and 3 ATP from NADH. So So if we complete oxidize the palmitate, and how many ATPs we can produce, so this kind of equation that you calculate, right? So 35 ATPs that can be produced from beta oxidation, just the beta oxidation, right? So 35 ATPs can be made from beta oxidation of palmitate fatty acids. But if 18 carbon fatty acid the number can be different, right? So just again, this just give you a sample how to, calculation. And then all this 8 acetyl-CoAs will be further oxidized, right, in the TCA cycles. So we need to add a TCA cycle, ATP generating from a TCA cycles. So again, eventually we will have 8 acetyl-CoAs enter the TCA cycles. So ATP numbers should times multiplied by 8, right? Does that make sense? So you know that one TCA cycles will produce 12 ATPs, right? Remember this? So 8 times 12, then is 96 ATPs. So 96 ATPs are produced from TCA cycles from 8 acetyl-CoA molecules. So final numbers will need to be added together, right? So 35 plus 96 ATPs, so the 131 ATPs can be produced from oxidizing one palmitate. And if you still remember that the first steps, the activation of fatty acids to fatty acyl-CoA, that you utilizes 2 ATPs, right? So 2 ATPs need to be subtracted, so the net yield of ATPs from palmitate oxidation is 129 ATPs, right? So if you compare to oxidation of glucose, the ATP production from oxidation of one molecule of glucose, still remember, only generate 30 to 38 ATPs, right, from glucose oxidation. So 129 versus 36 or 38 ATPs is much, much more, right? So oxidize one fatty acids produce much more ATPs than oxidizing one glucose. So take home message, because fatty acids have more carbons, right? And glucose have only 6 carbons. And these fatty acids may have 16, even more carbons. So take message, home message is molecules have more carbons and usually have more energies if they oxidize, right? Okay, now let's talk about oxidation of unsaturated fatty acids. So unsaturated fatty acids has a lot of double bonds, so beta oxidation cannot occur within the double bonds. So so first of all, for example, if we oxidize the linoleic-CoA, linoleic acid has 2 double bonds, right, at delta 9 and delta 12 position. So first, we go beta oxidation to break down the carbon from 2 and 8 position, right, and remove the 3 acetyl-CoA molecules and generating these molecules, right? Dieno-CoA. So because of the existence of double bond here, so the beta oxidation cannot, takes place here. So what we need to do here is to rearrange double bond and to make the single bond carbon available for beta oxidation. So rearrangement uses this enzyme called isomerase to rearrange the double bond. And this in the original molecules, the double bond is located at 3 and 4, right? Carbon 3 and 4 position, and after isomerization or rearrangement, the double bond is switched to 2 and 3 position, right? And to make a 4 and 5 carbons that are available for beta oxidation. So now you can further remove the 2 carbon unit from the acyl-CoA molecules and generate another acetyl-CoA molecules, right? And similarly, you can do more this kind of isomerization step, so reorganization double bond, and then go the beta oxidation, cleavage of acetyl-CoA molecule. So eventually, but compared to the the saturated fatty acids, the unsaturated fatty acids, again because of double bond, so you make less efficient to in terms of oxidation, so generate less acetyl-CoA molecules, right? All right. Okay, so any questions about, yeah?
Acetyl-CoA is only a process of saturated fat oxidation?
Yeah,
Or they just
right, yeah. Yeah.
In the third, in the slide before this one, in the third one, the processes that acetyl-CoA is taken out.
Mm-hmm, yeah,
It's just
right. Yeah, this, the last product, because of 2 double bonds they existed, so you can actually, do the beta oxidation. So they just stop. This will be the last
you stop
product,
when the double bond.
Hmm?
The oxidation stops when it finds a double bond.
Yeah, I mean,
The oxidation.
the oxidation, yeah.
It stops when it finds the double bond.
Oh, yes, right. They cannot do more oxidation, cleave the, the 2 carbon unit once it's, the double bond is there, right?
Does this process take more energy or less energy than oxidation and saturation?
I think it was less energy. But the cycle is the same, right? But just generate less energy because you generate less acetyl-CoA, right?
So
Yeah.
does it happen just as much as the other process with saturated fat, or does it just depend on fat in the
Yeah, I mean, usually our body utilize the saturated fatty acid for energy. I mean, we don't actually oxidize much the unsaturated fatty acid. So unsaturated fatty acid have a different function. We will talk about this like essential fatty acids later, right? So like palmitate actually is the most abundant, and we utilize a lot for energy. Okay, so now more question? No? Okay. So biosynthesis of ketone bodies or ketogenesis, right? So when our body does ketogenesis, do you guys know? In which physiological condition that ketogenesis occur? Fasting, Yes, fasting starvation, right? Um, and not just the fasted. I mean the long-term fasting, right? For example, we're fasting for longer than 2 days, and then the ketogenesis was very active. So this was under the physiological condition, right? Fasting. Um, but ketogenesis can also occur under the pathological conditions when we have very high levels of fatty acids in the circulation. For example, very, very long starvation, right? Or diabetes. So in diabetic patients, they will also have very high levels of fatty acids in the circulation. So liver picks up fatty acids and makes the ketone body in the liver. So again, the ketogenesis not only occur in the fasting physiological condition, but also can come kind of disease condition, right? The diabetic condition. So in which tissues ketogenesis occurs? Liver, yes, liver. So liver and mostly the ketogenic process takes place in the mitochondria, right? In the liver, in the mitochondria. This is just an overall picture of the ketogenesis. So fatty acids, here we just talk about the physiological condition, right? Fasting, starvation. We have a lot of fatty acids being released from adipose tissue, right? During the fasting, and the lipolysis process is activated. So the fatty acids, a lot of fatty acids go to the muscle and other tissue for directly oxidize, and you use for ATP production. But some of the fatty acids, like 1/3 of fatty acids, right, released from fat tissue, go to the liver for ketogenesis. Because of, so the reason I also wanted to ask why we need to make ketone bodies, why we cannot just use the fatty acids, right? Since fatty acids is so efficient, energy generator. Yes. Yes, great point. Yeah, thank you. Brain cannot use fatty acids because fatty acids is long, the larger molecules, right? Cannot pass through the blood-brain barrier. So, ketone body is much shorter, much smaller, the molecules, so be able to pick up by the brain and utilize in the brain. So, need to make a ketone bodies. And fatty acids in the liver, fatty acids be oxidized, right, through the beta oxidation and generate the acetyl-CoA. The majority of the acetyl-CoA in the liver being utilized for ketogenesis, right? So it's also kind of special in the liver. Most of the nutrients will not completely oxidize, partial oxidize, right? And here's an example. The fatty acids only just break down to the acetyl-CoA, and acetyl-CoA, majority of acetyl-CoA will not go into the TCA cycle for complete oxidation, rather actually go to the ketogenic pathway for making ketone bodies in the liver, right? And also other thing interesting that the liver makes ketone bodies, but they usually cannot utilize ketone bodies, right? So they release ketone body into circulation used by other tissues. So the reason why liver cannot use ketone bodies, and we will talk about this in the next slide, right? Okay, so ketone bodies basically have three compounds: the acetone and acetoacetate and beta hydroxybutyrate. Acetone basically is the byproduct, right? Can spontaneously decomposition of acetoacetate to the acetones. So acetones is excreted in the lungs, right? So that's probably is the reason, for diabetic patients, you sometimes you can smell kind of very special smell, right? This basically is caused by the exhale a lot of the ketones. So and acetone acetate and hydroxybutyrate, these are the most common ketone bodies in the body, and it's also relatively high levels, right, in the circulation compared to acetone. And these two ketone bodies can interconverted by the enzyme called hydroxybutyrate dehydrogenase, right? So ketogenesis. So let's start with the acyl-CoA fatty acid oxidation. So acyl-CoA can be oxidized by beta oxidation process, right? So the acetyl-CoA is the final product of the beta oxidation, but the second last product is the acetyl-CoA, right? And this acetyl-CoA together with acetyl-CoA form the hydroxyl methylglutarate-CoA, or HMG-CoA, right? So HMG-CoA can be produced from acetyl-CoA and acetyl-CoA, right? And this is enzyme HMG-CoA synthase. And then once the HMG-CoA is formed, and then the next step is the cleavage, right? So this HMG-CoA can be break down again into the acetoacetate, which is a ketone body, and the acetyl-CoA again. So acetyl-CoA can be recycled, right, for the next cycles of a HMG-CoA formation. So HMG-CoA lyase is the enzyme that break down the HMG-CoA into acetyl-CoA and the ketone bodies, right? So and acetoacetate can be converted to hydroxybutyrate, right, by these enzymes. So multiple steps, and again the acetyl-CoA is substrate or the starting molecules of the ketogenesis, right? And this acetyl-CoA majority or primarily is from the fatty acids beta oxidation, right? And this step is actually very similar to the cholesterol synthesis we will discuss later, right? Just try to remember the ketogenesis and cholesterol synthesis has some common steps, right? The cholesterol is also synthesized from acetyl-CoA, right? So the several initial steps are the same in both pathways. Okay. Let me just try to finish this slide. So this again is a big picture, right? Where these ketone bodies produce and which tissues it uses. Again, during the fasting, when we have high levels of fatty acids in the circulation, liver picks up and oxidize through the beta oxidation, generate the acetyl-CoA. So acetyl-CoA as the precursor or the starting molecules of ketogenesis, so synthesize ketone bodies, right? And ketone bodies made in the liver but cannot be used in the liver, but released into circulation, and the other tissue pick up ketone bodies, and ketone body can be break down back to the acetyl-CoA and utilized in the TC cycle for energy production in other tissues, right? So the tissue that utilize ketone bodies is multiple, but mainly brain and skeletal muscle, right? Okay, so any questions? Yeah.
What can you do in the blood rather than just produce acetone?
What kind?
What can ketone bodies do in the blood besides producing the acetone?
You mean this side?
In the second, the blood.
Oh, here?
Yeah.
The ketone body, what's your question? I'm
What
sorry.
can they do there in the blood normally?
Oh, in the blood?
The blood, yeah.
Well, they usually pick up by the tissues and use the energy.
So the blood is just a transportation method.
Right, right. Okay. Okay, so informal writing.